Hey, what's going on, Science 10s? It's Mr. Walton here. Today we're going to talk about the simplest ways that cells can take in nutrients and get rid of waste, and it's called passive transport. Before we really begin, we need to review a concept called diffusion. Now, diffusion is the net flow of particles down a concentration gradient, or from areas of high concentration to areas of low concentration. Let's take a look at diffusion in action. Here we have a simulation with two areas separated by a divider. On the left, we have some blue particles with a high concentration. On the right side, we have a completely empty space. We could call that area with low concentration. Notice that the blue particles are moving, but each one of them is moving with a completely random direction. What do you think will happen when I remove the barrier between them? If you said that the blue particles would start moving to the right, you would be mostly correct. When the divider is moved, the particles moving in random directions get spread out. And some of the particles start moving towards the right side. Not all of the particles are moving to the right, though. Do you see that particle right there? Let's watch this particle as it travels. As you can see, even though that most of the particles move to the area of low concentration when the barrier is removed, we still have movement of particles in all directions. Let's try this again, but we will start with particles on both sides of the divider. What do you think will happen to the blue particles when the divider is removed? As you can see, the blue particles still move from an area of high concentration to an area of low concentration. The same happened for the red particles as well. No extra energy was needed to allow this process to happen. So let's go ahead and illustrate what we just saw. We're going to draw a beaker or a container, and what we're going to do is add some particles on the upper left-hand side. Now, these particles in this beaker, let's say the beaker was filled with water, these particles in this beaker are going to be moving in random directions, and they will eventually, through no extra energy required, move in random directions, and they'll eventually fill in the entire container. So we will eventually get a uniform distribution because these particles are moving from an area of high concentration to an area of low concentration. And we should get an approximately equal distribution across the entire beaker. In general, substances can be moved into or out of the cell in two different ways. The first way is called passive transport. Passive transport doesn't really rely on any extra energy being used. Diffusion is the main driving force behind passive transport. Particles that undergo passive transport are always traveling from areas from high concentration to areas of low concentration, just like in the simulation we watched. Some molecules can diffuse straight across the membrane, like carbon dioxide and oxygen. These molecules are small and nonpolar, so they can pass through the nonpolar lipid tails of the phospholipid bilayer. Most molecules can't pass straight through the membrane, and they need transport proteins to facilitate their diffusion. We call this process facilitated diffusion. Notice that they're still traveling from an area of high concentration to an area of low concentration. Sometimes a cell needs to store large amounts of a substance inside or outside the cell. This requires the movement of particles against its concentration gradient. Because we're working against the flow of diffusion, this process will always require extra energy to be used up. Anytime energy is used to transport substances across the cell membrane, we call this process active transport. So let's summarize what we've talked about already. Under passive transport, no additional energy is required. Molecules or particles always move from high to low concentration. And I'm going to use 2 square brackets to represent concentration. So from high to low concentration. This, another way of saying that is with its concentration gradient. So like the natural flow of diffusion. Some examples of this are just simple diffusion and facilitated diffusion. Active transport requires energy. Specifically, it requires energy in the form of ATP. ATP stands for adenosine triphosphate, and you'll talk more about ATP when you go into Biology 20. So active transport requires additional energy input. It also transports things against its concentration gradient. So from low to high concentration, or again, against the concentration gradient. Some examples of this, as we'll see later, is the sodium-potassium pump. So that's a carrier protein that pumps sodium ions and potassium ions against their concentration gradient. So let's write this down just to be really explicit about it. Again, diffusion is the movement of particles from areas of high concentration to areas of low concentration. Molecules always travel down their concentration gradient. Diffusion rate can be controlled by adding energy and increasing molecular collisions. All the molecules of one type of particle diffuse independently of molecules of another type of particle. And we saw that in the simulation with the red and the blue particles. Diffusion requires no extra energy. Diffusion is a type of passive transport. So let's go ahead and let's draw a little bit of diffusion. So we're going to draw a plasma membrane. And it's a little bit difficult to draw a plasma membrane. I'm going to draw the phospholipid bilayer. Now I'm not going to draw the tails of every single one of them. I'm just going to draw some squiggly lines in between here just to show that this is a phospholipid bilayer. Now let's say this was the outside of a cell and this was the inside of a cell. Now, on the outside of a cell, typically you have a high concentration of O2. So you've got a high concentration of oxygen. On the inside of a cell, you typically have a high concentration of carbon dioxide. And so what happens, because these molecules are small and nonpolar, they can diffuse across this membrane and they can travel down their concentration gradient. So the oxygen molecules will travel inside the cell, or at least there will be a net movement inside the cell, and the carbon dioxide molecules will have a net movement outside of the cell. And that's good. Your cells don't want to build up with an excess of carbon dioxide. Your cells produce lots of carbon dioxide and they want to get rid of it. They use up oxygen and they want to get more of it. And so these gases are allowed to pass through via simple diffusion. We're going to do a diffusion experiment with a starch solution and iodine solution. The reason we're using starch and iodine is because when the starch and the iodine are allowed to react with each other, they make a dark purple color. This will allow us to see any mixing if it does occur. We will start by pouring some starch solution in a sealable bag and putting it in the beaker surrounded by a solution of iodine and water. Now, if the starch molecules can diffuse across the bag through the membrane, the solution in the beaker will start to turn purple. But if the iodine molecules in the beaker can diffuse through the bag or across the membrane, the solution inside the bag will start to turn purple. Let's watch and see which is the case. This video is a time lapse of about 14 hours. As you can see, the brown color of the water is starting to disappear in the beaker. We're starting to see some purple color in the corners of the bag appearing. I redid the experiment in a dark room again, and these are the results that I got. As you can see, there's no purple color in the beaker, so there's definitely no starch leaking out of the bag. But there is some purple color in the bag. That must mean that the iodine molecules can diffuse across the membrane of the bag. But if the starch molecules can't and the iodine can, when a membrane allows one thing to pass or some things to pass through it, but not others, we call that selectively permeable or semi-permeable. This is similar to the cell membrane, where the membrane is permeable to things like oxygen and carbon dioxide, but not permeable to things like potassium ions or sugar. Here's an example of what we mean by selectively permeable. At the top, we've got a high concentration of green particles, but no blue particles. At the bottom, we've got a high concentration of blue particles, but no green particles. Now, the membrane in between, so far, has no openings for anything. But when we open up the transport channels or the transport proteins for the blue particles, all of a sudden, the blue particles can diffuse across the membrane. So the membrane is permeable to the blue particles, but not permeable to the green particles. This membrane is selectively permeable. Notice that the blue particles are diffusing mostly from high concentration to low concentration, but every once in a while, one of the blue particles will move from low to high. We just have more particles moving from high to low. Facilitated diffusion is exactly the same thing as diffusion, but it's just for molecules that can't pass directly through the phospholipid bilayer. So the diffusion of a substance along its concentration gradient or with its concentration gradient, again, no energy is required. This time it uses a channel or a carrier protein to allow the substance to diffuse through the cell membrane. So we've got our phospholipid bilayer, but we're going to use a protein, and they have to be a transmembral protein because they have to be able to span the entire width of the phospholipid bilayer, the membrane. Okay, so we have our membrane. This is inside of the cell, and this is outside of the cell. And every once in a while, you'll get a buildup of certain nutrients, or you get a buildup of some waste products. And these products, you want them to be able to diffuse outside of the cell. Now, the problem is a lot of particles are either too big to be able to squeeze their way. They can't squeeze their way through the membrane itself or the phospholipid bilayer. But if there's an appropriate channel protein or a protein carrier, then these substances can diffuse from an area of high concentration to an area of low concentration through this protein channel. And this protein channel is very specific to certain molecules or certain substances. Now, one substance that requires a protein channel or facilitated diffusion is osmosis. Osmosis is the diffusion of water across a selectively or semi-permeable membrane. Now, water is a relatively large molecule. It's not really, but it's very polar. And because it's so polar, it is not allowed, or it cannot pass through this phospholipid bilayer all by itself. Now, water still passes from an area of high concentration to low concentration. Osmosis is just a different form of diffusion. So let's go ahead and let's draw. We're going to draw a selectively permeable membrane, and we're going to draw water, lots of water, on the left-hand side. And we're going to draw a relatively large amount of water on the right-hand side as well. But on the right-hand side is some dissolved solutes. Now, these solutes take up space. They take up space that could be occupied by the water. If we were to talk about the relative concentrations of water and solutes, here I'll just draw a legend over here. The blue is water and the red is a solute or something that's dissolved in the water. Now, the water has a really high concentration on the left-hand side. The water has a little bit lower concentration on the right-hand side. The solute has a really low concentration on the left-hand side, and it's got a relatively high concentration on the right-hand side. Hopefully this makes sense. So anytime you have high concentrations of solute, you also have low concentrations of water. It's a little bit backwards to think about this. Now, imagine this membrane was permeable to water. The membrane was permeable to water. What would you expect to happen? Water can pass through. It can pass through in either direction. But remember the diffusion of water, so it's always going from high to low concentration, right? So water is mostly going to pass through from the left side to right side. Yes, a little bit of it will pass from the right side to the left side, but mostly it will pass from the left side to the right side. Another way of thinking about it, and for me, this is a more powerful way of thinking about it. Solutes can be thought of as tiny little sponges. And wherever you have more solute, we've got more solute here on the right-hand side, they absorb or they pull in or they suck in more water. So you're going to see lots of sponges on the right-hand side. That means you're going to get lots of water moving that direction. We can see the difference between diffusion and osmosis in some YouTube experiments. Now, this is kind of ridiculous, but what we have here is we have a tube that's shaped like the letter U. We call it a U-tube. And so what's happening is we've got a membrane that's dividing the left-hand side and the right-hand side of this U-tube. Now, our membrane here is permeable to both sodium chloride and water. So let's go ahead and kind of draw in or fill in what's going on. On the left-hand side, we've got a solution of 2% sodium chloride. I'm going to draw that using 2 particles of solute to represent 2%. On the right-hand side, it's 8. So I'm going to draw 8 of these. So we've got 1, 2, 3, 4, 5. There we go, 8. So if the membrane is permeable to sodium chloride and to water, what's going to happen? Well, let's look at our solute. Our sodium chloride, or our solute, is going to diffuse from an area of high concentration to an area of low concentration. So our solutes will mostly pass to the left. Yes, a tiny amount will pass to the right, but it will eventually even out. Remember, diffusion causes the concentrations to even out. So what's going to happen? Well, our level of our water isn't going to change, and we're going to have an average concentration of 5% on both sides. It's just the average of the two. So hopefully you can see how if the membrane is permeable to basically everything, we just get everything evening out. That's diffusion. That's different, or a little bit different, to the next case. Here we've got a new U-tube, and the membrane here is only permeable to water. So we still have our 2% solution. We still have our 8% solution on the right-hand side. Now, our solute, our amount of solute cannot change on the left-hand side and the right-hand side. But what can change is the amount of water. Think to yourself, where are there more sponges? The solutes are sponges, and they like to attract water. And so, yes, we have a little bit of sponges on the left-hand side. That will pull a little bit of water that direction. But we have way more sponges on the right-hand side. So that's going to pull in way more water to the left or to the right-hand side. And so what happens is we still have, again, the salt. We have the same quantity of salt on both sides. 4, good. But the water is going to move inside this tube to the right-hand side. So the level of water is going to change. And that's because there's more sponges, there's more solutes on that right-hand side. And those solutes absorb water. They like to pull in water. They like to attract water. Another way to think about this is there was a lower concentration of water on the right-hand side, so now the concentrations have equaled out. Let's do another experiment. This time we're going to take some eggs and dissolve away their shells. We do this by setting a raw egg in vinegar for about 24 to 48 hours. As you can see, the vinegar is reacting with the eggshell, and the membrane of the egg is left intact. This naked egg is actually one giant cell. Remember, water can cross this membrane without adding any extra energy as long as we put it in a really high or really low concentration of water. On the left side, we're using distilled water. On the right side, we're using a mixture of water and molasses. Molasses is just a really concentrated mixture of sugar and some other dissolved solutes. After several hours, here's what the eggs look like. We can compare these eggs to a regular naked egg, and we see what happened to them after being placed in their respective solutions. So using what we learned from our egg experiment, what we're going to do is we're going to try and imagine or do a thought experiment about what would happen to our cells. Let's imagine our blood cells or something really important in our body. So our cells can be placed in different concentrations of solutions. What we're going to do, compare the solute concentration inside and outside of the cell in order to predict the movement of water. So here, in this first case, we have something called a hypotonic solution. Our cell is placed in a solution that has a lower solute concentration. This solution is hypotonic. Tonic meaning dissolved solutes, and hypo meaning not enough or lower. In the middle, we've got a solution where the concentration of solute on the inside and the outside is roughly the same. We call this isotonic. And in the third case, we've got a situation where the cell has been placed in a solution with a lot higher concentration of solutes. And so we call this being placed in a solution that's hypertonic. Hyper meaning too much. Think of hyperactive. If you're hyperactive, you have too much energy. If you've been placed in a solution that's hypertonic, it's got too much dissolved solids in it. So let's think about this. If you place a cell in a solution that's hypotonic, let's think about where the sponges are and what's going to happen. How many sponges are on the inside? Well, a fair amount. We're going to get a decent amount of water traveling into the cell. How many sponges are on the outside? Well, a tiny amount. We're going to get a tiny little amount of water going out of the cell. So where is the net? Where is the net movement? Inside the cell. In an isotonic solution, you've got a fair amount of solutes inside the cell, and you've got a fair amount of solutes outside the cell. And so the net movement, if you've got a bunch going in but also a bunch going out, the net movement is no movement. In a hypertonic solution, you've got, again, a fair amount of dissolved solids going on the inside, so they're absorbing water. But where do you have the most sponges? You've got way more sponges on the outside. So the net movement is you're going to get water moving out of the cell. One thing that can happen is that these cells in a hypotonic solution, if there's too much water moving in, the cell can get so big that it can burst. It can actually explode. In an isotonic solution, that's like a medium amount. It's a nice, happy amount where the cell isn't going to get any bigger and it's not going to get any smaller. But a hypertonic solution, the solutes on the outside of the cell are pulling water out, and what you end up with is a tiny little shriveled cell. And so that's exactly what we saw when we looked at our eggs. In one of our eggs, when it was placed in distilled water, it got huge. It was massive compared to what it started out with. In one of our eggs got a little bit smaller because the molasses pulled the water outside of the cell. Hopefully that makes sense, and hopefully the passive transport lesson wasn't too much information for you. Tomorrow, or in the next lesson, we're going to talk about active transport. Thanks. Have a great day.